In this video, we're going to look at 2 mosquito control strategies and some concerns that we may have with their implementation. The first strategy we're going to look at is the Oxitec OX513a genetically modified mosquito, where they have a tetracycline repressor so that the mosquitoes die if they don't have exposure to tetracycline, and the mosquitoes and the offspring would live if they do have exposure to tetracycline. This is similar conceptually to diluting the chances for a successful pairing by introducing sterile irradiated males. The first concern we'd have is with the separation of the male and female mosquitoes, because only the males are going to be released because the females bite and transmit disease. So as you can see in this picture, this is taken from Jove, the online video journal, of the process that's used to separate the male and female mosquitoes. You can watch the video and judge for yourselves what you think about this procedure. The second concern we have is that from the graph and table, this is from the article that's linked at the end, that there are other antibiotics that are more potent in relieving this transgene so that the mosquitoes would live than tetracycline. For example, doxycycline looks to be 100 times more effective at countering that transgene repressor than tetracycline itself. Some of the questions that we have about this strategy are, why wasn't a mixture looked at? That's what's found in the environment. They're not going to be released in the lab. They're going to be released into the environment, where a mixture of antibiotics that may have additive or synergistic effects in relieving the transgene. Second, what's the sorting accuracy? How many females are being released? Why wasn't the more powerful transgene nullifier, doxycycline, looked for in the environmental study? In addition, going to the theory of this strategy, why use a common antibiotic class in the first place? Wouldn't a novel chemical have been a better idea to be used as the repressor of the transgene? Is it possible that the mosquitoes, if they were to be exposed to water or animals that had certain levels of these tetracycline that relieve the transgene repression, that they would then survive in that environment? The second strategy we're going to look at uses lemongrass and a type of Trojan Horse to kill the mosquito larvae. Lemongrass essential oil is put within a yeast particle that is then put out for the mosquito larvae to feed on. Apparently they love the yeast, and after 3 to 4 days of feeding on it, they die. This is due to the constituents in the lemongrass essential oil. The link at the bottom of the slide is to a video where you can see and hear the research team talk about their discovery. So some of the questions we have on the lemongrass essential oil Trojan Horse strategy are, does it affect non-target species like tadpoles and fish? And if it does, is there a differing level of sensitivity so that the lemongrass essential oil could still be used at a level that would not affect these non-target species? Could a bait enclosure be designed so that only the mosquito larvae would be able to obtain access to the lemongrass essential oil Trojan Horse bomb? Another question is, what is the half-life, or how long does this lemongrass Trojan Horse retain its potency once it's put out into the environment? At the bottom of the slide, I've put a picture of some of the common lemongrass essential oil constituents. It is thought that the presence of multiple compounds makes it more difficult for the organisms to develop a resistance to the treatment. And here are some links to the sources that were used in this video.